Okay. So, looking at transcription, quick overview of all of gene expression. When we talk about gene expression, that just means taking DNA and turning it into something useful. In this case, we're thinking protein. So, 2 steps, transcription, translation, and because there are so many words that are the same for both, initiation, elongation, and termination, I want to spend a second and just re-clarify which are which. So, take 10 seconds for transcription and try to remember what initiation, elongation, and termination include. Remember, when you're thinking transcription, that's the production of mRNA. So what enzyme is going to be critical to all of this? Okay. So initiation happens when what? Louder. Once RNA polymerase binds to the promoter, what is that RNA polymerase going to do? Elongate. Good. It's just adding complementary base pairs. Reminder, and hopefully this is so well ingrained in you that you look at me and think I'm crazy for even bringing it up. But let's remember what the DNA and the RNA. So what are the 4 nucleotides in DNA? A, T, C, G. If your DNA is an A, what is the RNA going to have? If it's T, C, and G, good. Okay? All right. So the RNA polymerase is going to bind to the promoter, open up the DNA, add the complementary base pairs, and then termination happens when what? This is easy. Let's go easier. Right? RNA polymerase stops. That's going to cause the dissociation of the RNA, the DNA, and the mRNA. And it's going to be because of some type of termination sequence, but we'll look at that in more detail here in a sec. So at this point, we've taken the DNA. That's our blueprint. We photocopied it. Our photocopier here is the RNA polymerase, and the photocopied version, which is kind of like, just a photocopy of a few pages, that's going to be our mRNA. Translation is all about building whatever the blueprint says to build. In this case, we're making a polypeptide from the mRNA template. Tell me about initiation. What has to happen? Good. So my mRNA has to bind to the small ribosomal subunit. What else has to bind? The large ribosomal subunit, and one more thing. I think I heard it. What was it? tRNA, and that tRNA is special in that it has what attached to it already? Which amino acid? What does AUG always code for? MET. There we go. So we've got the tRNA MET. So this assembly kind of comes together, essentially the directions for what to build with a delivery boy for the first amino acid, as well as the construction site. They all come together. Elongation happens when the codon or anticodon on mRNA. Does mRNA have codons or anticodons? And what are they going to bind to? And what does the tRNA bring? Okay, so the codon on the mRNA binds to the anticodon on the tRNA, bringing the amino acid, and that process is just going to continue until you reach a raise your hand if you feel like you remembered most of that. Raise your hand if you feel like this was a worthwhile review, just to take another look at. Okay. All right. Yes?
What do you mean by stop codon
on the mRNA?
Okay.
Sorry. So remember, the mRNA is read in groups of 3. A lot of people are thinking transcription has something to do with codons. Do not use the word codon in transcription. There's nowhere that it really comes into play. Codons are only important for translation. Okay, mRNA stands for, okay, so this is basically our photocopied DNA. tRNA stands for transfer. And what are the 2 parts on the tRNA that you must be familiar with? On the tRNA. The amino acid binding site and the, so we've got an anticodon and an amino acid binding site. When we get to the rRNA, R stands for, and the 2 parts there are small and large. Clarification here, the ribosome is made out of 2 things, rRNA and proteins, okay? But the rRNA is ribosomal RNA. It just has to work with proteins. Okay, there's our background. I promise that's going to be the most writing that you'll have to do. So a lot of information that should be all reviewed. This is nothing new, but we need to really kind of hone in on some of these areas and make sure that we add the details that we need now. Okay? All right, what is this picture showing you? And the central dogma says that DNA codes for mRNA, and what process is that again? And that mRNA does translation to make, give me a better word than protein, polypeptide. Okay, polypeptide has to be then folded to make the proteins. Quick review again, remember there's 4 levels of protein structure. The mRNA dictates that first level, which is just the amino acid sequence. That's our polypeptide. But then it has to be folded into those alpha helices or those beta pleated sheets, and then have longer ranging folding patterns and maybe interact with other polypeptides to make the overall protein. Raise your hand if you remembered all that part. Okay, good. Okay, again, quick review, make sure we're solid here. The template strand is the one that is going to be used to make the mRNA. The other strand is the coding strand. It is called the coding strand because it has the same code as the mRNA. For example, wherever there's a G, there's a G. The T's obviously are now U's, which gets a little bit confusing, but there's an A, there's a C, etc., etc., etc. So, mRNA is read in pairs of 3 or sets of 3 called the codon. Each codon codes for 1 amino acid. Nothing new there. We talked about the genetic code being redundant and nearly universal. What does that redundancy refer to? What's that?
It always starts with the same.
Not necessarily. That's true, but that's not the redundant part of it. I'll give you a hint. In this box is a prime example of redundancy.
Different orders of nucleotides can code for different the same amounts.
Perfect. So we can have different codons, different triplet pairs, coding for the same amino acid. So for example, if you had CU anything, it'll still code for leucine. It doesn't matter if that last or if that third base pair is U, C, A, or G. So it is redundant and nearly universal. What does that piece mean? Universal genetic code. Oops. Hope that stays up. Nearly universal. What is this a picture of? Come on, you guys, you got this. It's a piggy. Tell me something interesting about this pig. Okay, this pig, it is not just like a weird filter on the camera or anything like that. Its nose and its paws are glowing yellow. And so the reason it's doing that is because the genes that came from, I think this one is a jellyfish maybe, the genes from that jellyfish were inserted into the pig as a embryo, and those genes get expressed. because it's the same code that works for bacteria as works for humans, which means that you can take any gene and it's going to do transcription and translation and use the same mRNAs, tRNAs, etc., to be able to be turned into a polypeptide. So this is a happy pig. Doesn't he just look happy? He's even got little teeth up in there, like, okay, whatever. Okay, so this part on the left, that's just review. The blue stuff is DNA, the red is the mRNA, and the purple is, okay, so far so familiar. First step is trans fact, second step is, okay, today what we're going to do is look at the connection between structure and function. The fact that this nucleus is here means that this process is going to need to be a little bit more complex. Okay, so we're going to still look at transcription, but now instead of DNA making RNA, it's going to make like a rough version of RNA that we're going to refer to as pre-RNA. Right? It's not finalized. It's not ready to go out onto the field to make a building. And so that pre-mRNA must be processed. That's going to make your final mRNA, and that must then leave the nucleus to be translated. Okay. Structure, function. Why is it wonderful to have a nucleus? What is so good about the nucleus? Okay, think of it this way. What organisms are more complex? Prokaryotes or eukaryotes? Why? Okay, they have a nucleus, but that's what allows their complexity to occur. How? What's that? That's going to be really important. The fact that we have this membrane-bound system of organelles allows you to keep things in separate areas. And so the fact that we have our DNA separated from the rest of the cytoplasm means that this DNA is protected. It means that you can concentrate all of the enzymes needed for transcription in this one area with the DNA. But it also means that now we can't just do transcription and translation at the same time. Doesn't work anymore. If you're a bacteria, as soon as you start making the mRNA, polypeptides start getting made. Bada bing, bada boom, you're done. In eukaryotes, you got to keep these processes separate, because now you've got transcription happening where the RNA polymerase is, translation happening where the ribosomes are, but these two areas are separated by the nuclear membrane. Okay, so let's review quickly prokaryotic transcription, and this is the part where I give you a big, ginormous apology. When we went over prokaryotes, I had mentioned transcription factors. No, I was mega wrong, and I apologize. There are not transcription factors for prokaryotic cells. So what's going to happen is the RNA polymerase is going to bind directly to the promoter. That is initiation. Elongation happens, building the new strand in the 5' to 3' direction. And then a termination sequence is transcribed. That allows everything to break apart. Okay? Not too bad. Let's compare that quickly now to transcription in eukaryotes. For starters, the promoter is really similar. It's still upstream. So if this is our start point, we would call this +1. That's the first nucleotide that's going to get transcribed. So we're still looking upstream, like -10, -35-ish, in that range. But now we have this element right here, and it's a very, very highly conserved element that is found in almost all eukaryotic genes. A lot of eukaryotic genes. There's weird exceptions. But this is called the Tata box. Why do you think it's called the Tata box? Because there's a whole bunch of T's and A's, and for some reason scientists went against calling it the at box, which I think would have been more appropriate, but you know, calling it the Tata box is fun. So we've got this Tata box in as part of the promoter, and that is going to be where transcription factors bind. Transcription factors are going to bind to the Tata box in the promoter, and then and only then will RNA polymerase be able to bind. So we have essentially made this transcription initiation complex, this big old chunk of all kinds of proteins and the polymerase and the Tata box itself. Yeah, Anna.
So is the initiation complex like the last picture with the RNA polymerase?
Yes. So this is all part of the initiation complex. Right? So this entire picture on the right is just showing initiation. So that's different than what we saw with prokaryotes. The promoter's different. It doesn't have the TATA element. The transcription factors are not needed. The polymerase can bind directly to the promoter. Elongation is exactly identical, nothing special there. We're still building in the 5' to 3' direction. And now when we get to termination, instead of a termination sequence, we've got a polyadenylation sequence, which is just a fancy way of saying a whole bunch of A's. So that's a sequence that is going to tell the RNA polymerase that it can fall off, but it is also going to be involved later on in further processing of the mRNA. Okay, so if we take a quick look here, what are the 3 steps of transcription? Initiation. Okay. Initiation in eukaryotes is different. It's got transcription factors, the Tata box. Elongation is the same. What's different about termination? It ends at a polyadenylation. So just the process of transcription itself is just a little bit different. It's a consequence of evolution. You don't need to necessarily think of like, well, why would it be that way? It just kind of is. The RNA processing, though, that's completely new. That's something that is not seen in procs. So let's go ahead and look at what happens. Again, we're thinking about things like structure and function. So the RNA has to be able to be moved out of the nucleus without being degraded. There are hydrolytic enzymes that would otherwise break down nucleic acids. Why might that be? Why would you want to break down nucleic acids? If you are a cell, why would you want to make sure that there are no nucleic acids in your cell, in you? Because that's usually a sign of invaders, some kind of pathogen. So this is really great for our immune systems to be able to make sure that we don't get viruses. Also, the ribosomes are different on prokaryotes than in eukaryotes. So eukaryotic ribosomes need to have some things happening on the mRNA to be able to bind. Okay. So the 3 modifications that we're worried about here. If I'm looking at this RNA processing, there are 3 things that are going to happen. Something at the 5' end, something at the 3' end, and some splicing. Let's go ahead and look at what those are. At the 5' end, we're going to add a cap. So let's go ahead and also review what the 5' end is in reference to. This is one nucleotide. Up here, if you're looking in the top right-hand corner. Okay? I've got my 5-carbon sugar in the middle, my nitrogenous base kind of over on the right, and a phosphate group up on the top. If I take a look at my 5-carbon sugar, do we remember how to number this? said it 1,000 times. Let's make it 1,001. 1, 2, 3, 4, 5. This is the 5' end because it has the phosphate attached, 5 and phosphate, and then there's nothing past that. Now this technically down here should not have a phosphate, and that makes the 1, 2, 3 carbon available, so this is the 3' end. So on the 5' end, we do what is called a 5' cap. All eukaryotes do this. We're just going to take a couple extra phosphates and a modified guanine, and it just kind of gets put on the other end. Okay, so ribosomes are going to recognize that 5' cap and bind there. At the other end, at the 3 end, we're just going to add a whole bunch of A's. And so what do those things do? The 5' cap is going to be important for ribosome binding, and both of them are going to be important for export out of the nucleus and preventing any kind of breakdown. Okay. How are we doing so far? You're eerily quiet. What's up?
I don't know. I'm just getting confused of like where like the difference between the eukaryotic
Okay, so kind of if you take a look at this one, this is going to really highlight all of those main differences between the procs and the euks. All of this is just for eukaryotes. So if we were to take a look at prokaryotes, they don't have transcription factors. They don't have the ta-ta box as part of the promoter. Instead, what they're doing is they're going to be adding directly onto, hey, I'm like 2 minutes away. Okay. So they're going to be adding directly onto the, where was my train of thought? Promoter. Okay. Elongation happens the same. Termination happens because of a termination sequence instead of the polyadenylation sequence. So not too much is terribly different in transcription, but the RNA processing is going to be the big one. Okay? All right. If there's anyone in here that is thinking about going into bioengineering or any kind of biotechnology, this is where your money's going to be made. This is the cutting edge research. So, what is a nematode? It's a little worm-like guy, kind of looks like this. Many of you have probably had nematode parasites at some point and not even known it. Okay, all of you were born before 2003. Please tell me I'm accurate on that. Okay, so in your lifetime, the Human Genome Project was completed. The Human Genome mapped out every gene in humans. And you know what it found out? That you all, as phenomenal as you are, have the same number of genes as this little worm here. How can that be? You are complex, you are awesome, you are capable of thought. And this dude just kind of eats and makes sperm all day. That's all he does. Right? That is, okay, well, as a teenager, maybe you're there too. But why is it that you have the same number of genes as this guy? Why do you have the same number of genes as a fruit fly? That's ridiculous. And the answer comes down to the fact that your genes do not follow the 1 gene, 1 polypeptide rule. In fact, higher eukaryotes have a lot of what is called splicing. So splicing is basically taking the piece of DNA, turning it into mRNA, and then cutting out chunks of it. So the introns are the parts that interrupt, and the exons are the parts that get re-spliced together and get expressed. So you can have these big chunks of mRNA that get cut out, thrown out, not used. And that's going to allow for your genes to make all kinds of proteins. So this is going to be done by an enzyme complex called the spliceosome, which, if that isn't fun to say, this right here, the small nuclear ribonucleoproteins, which you do not need to know the full name of, it's just interesting because you know what scientists call them? Snurps. I'm 12. Okay. So, splice this guy up in a whole bunch of different ways, and I almost think of it like Lincoln logs or like those like connects. Depending on how you splice stuff together, you can make a lot of really cool different genes that have proteins that do all kinds of different things that you wouldn't have otherwise expected. Okay? All right.